Okay. All right, can you guys hear me? Let me
Yes.
plug in my headphone. I guess I should have asked now. Now can you hear me?
Yes.
Great. All right, are there any questions before we begin? If there's no questions, I'm going to go ahead and get started. You should be able to see my screen now.
Yes, it's visible.
Great. So today is June 2nd, and we are supposed to be working on chapter 13, viruses, viruses, and prions. We might finish that. We're supposed to finish that today. Once again, there is no lab. You should be working on your unknown project. Let me remind you, the unknown project is due not this coming Saturday, but a week after this Saturday. So when is that? Saturday, June 11th. And the unknown notes page will close Friday, June 10th. Any questions about any of this? There will be no lab today, but I will be logging in from 6:30 to 6:45 to answer questions. Don't think we need this. Trouble is if I close this, let's go ahead and, oh, I meant to have that open. Let me get chapter 13 open. Well, that's opening. Let me close that. Ah, that's not where we were. That's something I'd change. Where were we? You know, we talked about animal viruses. So we had talked about a multiplication of a DNA virus, and they need to reproduce both their genome as well as their proteins, the proteins of the capsid. And then those get together to make the new virions. So let's talk about the multiplication of a DNA virus in a little more detail. Let me blow this slide up. So initially, a virus will infect a cell, and we're talking about a DNA animal virus, but a DNA human virus specifically. And the virus attaches to the host cell. We did mention that there are ligands or adhesion molecules on the virus that bind to receptors on the host cell. And both of these molecules, both the adhesions as well as the receptors, tend to be glycoproteins. The virus then comes into the cell, and we talked about that. It can be by fusion of the viral envelope and the cell membrane, or it can be brought in by endocytosis. The virion enters the cell and its DNA is encoded. We're not going to talk about how that's done, but the virus separates and the DNA is then released. The viral DNA is then transcribed to make viral protein, and that'll be largely be the capsid proteins. And the viral DNA will be transcribed to make viral messenger RNA. The viral messenger RNA will then move into the cytoplasm, shown here. And then the viral messenger RNA will be translated to make viral proteins, which are the capsid proteins mainly. And then the capsid proteins will get together with the newly replicated viral DNA, which, where do they have that shown here? Right here, I guess. Viral DNA is replicated right there. Oh, there it is. One DNA is made into 3 molecules of viral DNA. And then the viral DNA will get together with the viral capsids to make the new virion. The cells are then released mostly by budding or something similar to budding, or they will lyse the cell, and then the lytic virus will be released. And then the virus will be outside the cell and it can attach to another cell. That's the life cycle of a DNA virus. Any questions about that? If not, let's turn to the RNA viruses. Let me blow this up. There are, I think, 15 families of RNA viruses that cause human infections. You don't need to memorize this table. We will talk only about a few of the families listed here. We'll talk about the Picornaviridae a little bit. The herpes viridae, we've already talked a little bit about. I don't think we talk about these. I'll talk a few slides about Coronaviridae, and then we'll talk a little bit about retroviruses, Retroviridae. I don't think we talk about, oh, we'll talk a little bit about Orthomyxoviridae. Anything else I need to talk about that?
Dr. Volpe,
What?
would we get tested on this slide?
Yeah, I'm not going to test you on, for example, asking a question like, is herpes viridae a naked virus or an envelope virus? Okay, I won't ask questions like that. I won't obviously ask, is the herpes viridae icosahedral or is it helical? Okay, I'm not going to ask you about the size either. If we talk about the virus, then I expect you to know the viral name and what disease it causes. I think we do talk about polio slightly, and we talk about rhinoviruses causing the human cold. And I don't remember if we talk about hepatitis A or not. These are picornal viruses. And so if I talk about it, you should know it. But like, I can't even read that, sorry. Let's move, here's one. Astrovirus causing gastroenteritis. I'm not going to talk about that, so I'm not going to quiz you on any of that. Okay? I don't think we talk about rubella being caused by the ruby virus, which is a toga viridae. I don't believe we talk about that. But if I talk about it, like COVID-19 belongs to family Coronaviridae, I do talk about that. And it causes, gosh, I'm not sure what the disease is called. We'll call it COVID-19, that disease. We'll talk about retroviruses, and you should know that HIV causes AIDS. Okay, so we will talk about that, but you don't need to know anything from this slide because everything we'll talk about will be in another slide. Any questions about that? The important part here, and probably the only thing you need to know, is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. There are 15 families of RNA viruses that cause human disease. That's probably the only thing you need to know from this slide. I'm just introducing the RNA viruses here. The RNA viruses contain RNA. We're only talking about RNA animal viruses, but realize there are other RNA viruses, and they affect the kingdom that the virus infects. Like there are plant RNA viruses that only infect plant cells. And these are just showing you some of the RNA viruses, not terribly important. The multiplication of RNA viruses is essentially similar for DNA viruses, but understand there are some differences due to having RNA as the genome instead of DNA. Viral replication generally occurs in the cytoplasm. So that's a rule of thumb. If it's an RNA virus, then it generally replicates in the cytoplasm. There is one exception to this rule of thumb, and that is the retroviruses are an RNA virus, but they replicate in the nucleus. And we'll talk a little bit more about retroviruses. When we're talking about single-stranded RNA virus, it's important to realize that there are two types. There's the positive strand, single-strand RNA, animal virus, and there's the negative strand, single-strand RNA, animal virus. The positive strand can be read by the ribosome. The negative strand cannot be read by the ribosome. And what it is it's the corollary to the positive strand. I think we mentioned this, a DNA virus, if it's double-stranded, to be replicated, all you need to do is separate the strands and then use each template to build the new strand. For an RNA virus, it obviously doesn't replicate that way. If it's a positive strand RNA, it replicates by making the negative strand RNA and then using the negative strand as a template to reproduce more of the genomic strand, which is the positive strand, single strand RNA. Any question about that? Now remember, the positive strand RNA may be used as a messenger RNA, in which case it can be translated into proteins. For the negative strand genome viruses, negative strand RNA genome viruses, to replicate, they have to make a positive strand RNA, single strand molecule, and then they use that positive strand as a template to make more of the genomic RNA strand. The negative strand RNA cannot be read by the ribosome, so it is not translated into protein. How the negative strand RNA viruses make their proteins is they make a positive strand RNA, and then they can use that positive strand RNA as a messenger RNA to be read by the ribosomes in protein translation. Any question about any of this? All right, so if we have a positive strand RNA genome, which is actually shown here, sorry, I should have started up there. We replicate that by making the negative strand. Let me blow this up. Talking about the positive strand. So we make the negative strand. To make more of the positive strand, we use the negative strand as a template to make more of the positive strand. And then the positive strand can be used as messenger RNA to make proteins. And then the proteins get together with the positive strand RNA to make the new virions. For the negative strand genome, we make the positive strand RNA and then use the positive strand as a template to make more of the negative strand RNA genome of the virus. And then we use the positive strand RNA to make the capsid proteins. And then the capsid proteins get together with the replicated negative strand RNA to make the new progeny virions. So this is an infection with a positive single strand RNA virus. The virus attaches to a host cell. The virus is brought into the cell either by endocytosis or fusion of the envelope to the cell membrane, assuming the virus has an envelope. And the RNA is unpackaged and it replicates in the cytoplasm right here, and it makes the negative strand. And then the negative strand is used as a template to make more of the positive strand. The positive strand is then used to translate proteins. Capsid proteins get together with the positive strand RNA to make the new virions. And then this virus either buds out of the cell, kind of shown here, or if it's a lytic virus, it lyses the cell, releasing the newly replicated virions. Any question about virus multiplication in a positive strand RNA virus? The negative strand is similar. The virus binds to the cell. It's brought inside the cell. The viral genome is then uncoded from the virus. The viral genome, which is negative, is then makes the positive strand and uses the positive strand to make more of the negative strand viral genome. The positive strand, shown here with this, is then translated to make the viral capsid proteins. The viral capsid proteins get together with the negative strand, newly replicated viral genome to make the new virions. And then the new virions either bud out of the cell or something similar to that. And if they're a lytic virus, they lyse the cell, releasing the newly replicated virions. Any question about viral multiplication? All right, let's move on. So let's talk a little bit about retroviruses. The family Retroviridae, discussed as retroviruses, includes the lentiviruses, which are viruses that AIDS is a family member to, as well as a feline leukemia virus, which causes leukemia in cats, which causes symptoms similar to AIDS in cats, but it's actually leukemia. The leukemia weakens their immune system, and then the cat gets sick from many different things and eventually dies. The cat kind of wastes away, similar to AIDS. Any question about any of that? Retroviruses are unique in that they are a RNA virus, but they do not replicate in the cytoplasm. They replicate in the nucleus. And they're also unique for another reason, and that is retroviruses within the virus itself contain an enzyme that we call, shown right here, right there, the purple one, or pinkish purple. That we call this enzyme reverse transcriptase. And this enzyme is encoded by a gene in the viral genome, which is RNA. Any question about any of that? Okay. Something that you can see in this picture is retroviruses actually have two strands. I don't have it here, but there are two positive strands within the virus. So it is a positive single strand RNA virus. And you don't need to know this, but there's actually two strands that are positive in the virus. The important thing that you should know is within that strand of RNA, there is a gene for reverse transcriptase. Reverse transcriptase is unique because it is an enzyme which takes RNA, which in this case is the viral genome, but it can be any positive single strand RNA. And most of the time, this will be messenger RNA. Obviously, for the virus, it's the viral positive single strand RNA. A reverse transcriptase can take this. Let me get an arrow here. Well, I don't see the arrow. Why is that? It should be showing. I'll try it again. Well, that doesn't look like an arrow to me. Ah, there, I finally got the arrow, but it's not facing the right way. How do I rotate that? I don't see a, oh, there we go. Rotate left, I think. There we go. Let's try and delete this. Sorry, I'm having a little trouble with my paint here for some reason. I'll just leave that block there. I got it. Let's go the select, cut, paste. Bring it. Oh, no, I didn't want that. Having trouble here. Lost part of the arrow there, but that's all right. There we go, right there. So the messenger RNA, and this is by reverse transcriptase. And I'm just going to go RT for reverse transcriptase. It makes single strand DNA. Oops, a little bit of a doohickey there. So this is single strand DNA. And then the reverse transcriptase, let's see if I can copy this. Oh, I didn't get the RT. Oh, fudge. Having a hard time grabbing the thing. Oh, let's make another one. And then makes double-stranded DNA. So the reverse transcriptase can take single strand, positive strand RNA to make single strand DNA. And then the reverse transcriptase can take a single strand of DNA to make 2 strands of DNA. We're just going to abbreviate that as double strand DNA. Any questions about that? This double strand DNA then moves into the nucleus. Let's see if I can copy that again. Nope. There we go. Moves into the nucleus and then integrates. I don't know how to draw that, so we'll try and do it with a different color. But then integrates. What the heck is that? There it is. But then integrates into a viral chromosome. And we'll make this a human chromosome. And I guess we'll make it the X chromosome. Okay. And the integration of the retrovirus kind of happens at random. That can be at the end of the chromosome. It can be in the middle of the chromosome. It can be anywhere in the chromosome. And it can be any chromosome. Any questions about any of that? So that's how reverse transcriptase works. And that's how the retrovirus works. Now, what the virus does is to replicate itself. The virus will, see if I can get this one to work this way. There we go. The viral genome will transcribe, making viral messenger RNA. And this is positive strand, that should be a little lowercase. And it's single strand too, so I guess I should go like that. This is a positive strand. Oh, that's not the right button. And that's the viral genome. And then that viral genome can be used to translate viral proteins. And this happens in the cytoplasm. So the viral RNA will move into the cytoplasm to make the viral proteins. And then the viral proteins, let's get this arrow, we'll get together with the viral genome. Let's just write that. And this is RNA, single strand, positive strand. I got that a little bit too far. And that'll make the new virion, which we're just going to say another box. New virions. And this is a lentiviruses as well as retroviruses have an envelope, and so they will bud out of the cell. So they replicate by budding. They follow the budding life cycle. And it depends on the species. Some budding viruses, as you remember, bud out of the cell membrane. That's probably most of them. But some bud out of the nuclear membrane, and some bud out of the membrane of the ER, the endoplasmic reticulum. Any question about the life cycle of a retrovirus? So you see, our retroviruses have a unique life cycle, and they have a DNA intermediate, which actually does integrate into the host cell that it infected, into the host cell's DNA. And that integration can happen essentially anywhere, along any chromosome in any position. And once that happens, for the lifetime of this cell, it will carry the retrovirus genome within the cell's genome. Any question about any of that? Well, here's a question for you. There are two ways that the retrovirus can replicate. One way is shown here, where it buds out of the cell, and I guess what we should do there is to complete the life cycle. It then starts over again. So that's one way that the retrovirus life cycle can happen. What's the other way that the retrovirus can reproduce? Come on, somebody's got to know. Nobody's going to even take a guess? What about a lysogenic bacteriophage? What were the two ways that it could reproduce? Is anyone there? I can't see anyone. I'm going to stop the share here. Yep, there's people here.
Does that inject the virions directly into the cell?
It doesn't inject the virion into the cell. Animal viruses have an easier time getting into the cell than bacterial viruses that we call bacteriophages, because animal cells do not have a cell wall. So animal viruses get into the cell either by fusion of the viral envelope to the cell membrane, and that'll dump the virus into the cell, or the virus can get into the cell by endocytosis. The second way a retrovirus can replicate is this cell containing the, excuse me here, containing the viral genome in the host chromosome. This cell can replicate. Let's get this arrow. And if that cell replicates, oops, that's the wrong arrow, but it's close enough. If this cell replicates, then we'll get two cells with the copy of the retrovirus integrated into the genome of the cell. So when this cell replicates, it will replicate the viral genome as well as the chromosomal DNA. All right. In animal cells, that's not a big way that viruses replicate. Like AIDS, for example. Theoretically, this could happen. but the virus is much more likely to replicate by budding. And I'd have to think about a virus to think of one that might replicate when the cell replicates. There are no human retroviruses that are functional in the human genome. We have 5 or 6 viruses in the retroviruses in the human genome, but all of them are mutated. So the retrovirus is non-functional and cannot replicate. But in the mouse genome, they have a retrovirus, which is in their genome. And when the mouse father gets together with the mouse mother, they can have a offspring, which will be replicating the retrovirus. Does that make sense? So in humans, there's no known case of it, meaning we do have retroviruses, but they're mutated and non-functional. They don't replicate in humans. But in a mouse, there is a live retrovirus that can replicate, and it's in the genome. And so that retrovirus is passed from mother or father or both to the offspring. All right, let's close that and move on to the next slide. I've already mentioned that retroviruses are positive single-strand RNA that contain reverse transcriptase. And first it makes single-strand DNA, and then the reverse transcriptase makes double-strand DNA. That double-strand DNA can be transcribed to make viral messenger RNA, which can be translated into the capsid proteins of the virus. And then the double-strand DNA can be replicated to make positive single-strand RNA, which is the viral genome. And then the viral genome gets together with the capsid proteins to form the newly released progeny viruses, which I've actually already talked about here. The retrovirus gets inside attaches to the cell, gets inside the cell, makes the viral DNA. The viral DNA integrates into a host chromosome. Oops, too far. The purplish, the pink purple, as opposed to the bluish purple, which is the rest of the host chromosome, making the viral genome. The viral genome is positive strand RNA, so it can be used by the ribosome to make viral proteins. And then they get together to form the new virus, which will bud out of the cell shown here. And the virion will come up to a membrane that has viral proteins in it, and then bud out of that membrane, ripping off a piece of it to form the envelope of the retroviruses. Any question about any of that? All right, any question about retroviruses? No question about retroviruses? All right, let's move on to coronaviruses. And you'll note that coronaviruses is actually the picture for my website. Oh, here's it. So that little picture for our course is actually a coronavirus. And I chose that just because I thought the picture was neat many, many years ago, maybe 10, 15 years ago, long before COVID-19 was around. And then I kept it because, well, it's coronavirus. I'm now talking about coronavirus. Coronaviruses is a family of RNA viruses. The family has a single positive strand, meaning a single strand, positive strand RNA virus. It is enveloped, and that's the envelope you're seeing here with the spikes. Obviously, in the coronavirus family, we have COVID-19, but coronaviruses have been known to humans for a long time. They're actually the second most common cause of the cold. So coronaviruses, there are species of them that cause the human cold, and they're the second most common cause of the human cold. The most common cause of the human cold are rhinoviruses in the family Picornaviridae. Okay, so it's the most common cause of the human cold. But coronaviruses are the second most common cause, meaning the second family. And then the third family we briefly talked about last time, adenoviruses, Adenoviridae, is the third family of viruses which can cause the human cold. Coronaviruses, besides causing the human colds, which I should mention that coronaviruses, when they're causing the human cold, they tend to cause colds in children. As adults, maybe because we've been exposed to them, we don't tend to get colds from coronaviruses. These are colds that children are more likely to get, although adults can get the cold from coronaviruses. Coronaviruses also cause, obviously, COVID-19, SARS and MERS. SARS stands for Severe Acute Respiratory Syndrome. MERS stands for Middle East Respiratory Syndrome. Unlike the coronavirus that cause human colds, SARS, MERS, and COVID-19 all have a mortality rate. The most mortal, meaning the one with the highest mortality, is MERS, but fortunately MERS does not spread from people to people. MERS usually spreads from camels to people. And the mortality rate of MERS is around 36%. SARS has a less high mortality rate. The mortality rate is around 10%. If you remember, SARS was a disease, I think it was 2003, the first time it appeared. And I don't remember, it's appeared one other time. Both times it appeared in China from the food markets. The Chinese have a whole bunch of animals in their food market because they eat strange animals like spiny anteaters, and I don't know what all the other animals are. Let's just say lizards and anteaters and whatnot. I don't know what they all have, okay? And they have these animals live, which can contain viruses. And we have 3 incidents now where we think from these animal food markets in China, we had 2 cases of SARS coming out of the food markets, as well as we think this is where COVID-19 came from. So we've had 3 incidences of this. And the Chinese, after 3 incidents, have said, you know, this isn't safe. We're getting lethal viruses coming out of our food market. We should clean up these food markets and not allow all these animals in close population with the people. And it's believed that the animals contained the virus, and then because the people were in close contact with the animals, the virus moved out of the animal and into the people. Initially, the first time, it may have been only one person who came down with the virus, but then that person spread the virus to other people. Any question about any of that? COVID-19 is less lethal than either MERS or SARS. The lethality rate is someplace between 0.1% and 3%. We'll talk about what it actually is in the United States. They don't know the exact mortality rate, although they should have a pretty good estimate because we've studied it now for many years, at least over 2. The trouble with the mortality rate is that different strains have a different mortality rate. The problem with COVID-19 is it spreads much more easily than the flu does. So it spreads much more easily than the flu. And one person is likely to infect between 1 and 2.5 other people, which is why we're having such a hard time controlling it. Once the pandemic started or the epidemic started, it just spreads from one person to another. In the United States, there's over 1 million deaths now from COVID-19. And as of late May, the number of the US population who have become infected with COVID-19 is actually 60%. So a large majority of the population have been infected with COVID-19. And the sad thing is that once you've had COVID-19, you can get it again. Your immunity lasts 6 months to a year at most, especially if you get the viral disease. Even if you get vaccinated, the immunity won't last longer probably than about a year. So we'll need to give booster shots at least once a year. A problem with COVID-19 is different strains evolve. Do I have a slide of that? I think I've got a different side of that. So we'll talk about the different strains, yeah, on that slide there. And when one strain hits you, that will not protect you to another strain. Is that clear? It depends how similar the strains are. Like if you got infected with, is it AD1 for Omicron or Omicron? It's pronounced both ways, and I'm not sure how you pronounce that. The strain going around now, I think it's AD2. You get some protection from Omicron or Omicron, but the protection isn't 100%. And I don't think they know how much protection you get, because the new strain is just too new. All right, any question about any of that? All right, COVID-19, I didn't mention where the name comes from. Let's go back here. COVID-19 is a severe acute respiratory syndrome 2. meaning SARS or SARS-1 is SARS, severe acute respiratory syndrome. COVID-19 is severe acute respiratory syndrome 2, but that name is so similar to SARS that not even the scientists use it. Most people call it COVID-19. It's called COVID-19 as an abbreviation, CO or COV for coronavirus. And then it was ID'd, identified for ID in 2019. That's where the 19 comes from. So the name is an abbreviation for coronavirus identified in 2019. Any question about that? And there is the correct name for COVID-19, severe acute respiratory syndrome coronavirus 2, or SARS coronavirus 2. COVID-19 is closely related to bat coronaviruses. It likely originated in bats. When we sequence the coronavirus, the first one that was circulating in the human population, we find that it is a recombination of 2 different bat coronaviruses that join together, and those 2 bat coronaviruses then recombine with the pangolin coronavirus. A pangolin is a spiny anteater. The pangolin genome is only about 10%, so what is it? 45% belongs to one bat coronavirus, and about 45% to the other coronavirus. Only about 10% of the genome comes from the pangolin coronavirus, meaning that's a coronavirus that infects pangolins. And the 10% actually codes for the spikes on the coronavirus. There are different strains of the COVID-19. The strains are mutations of the original, and they have different names. The original is just called the original coronavirus 19 or COVID-19. There was alpha, there was beta, there was gamma, there was delta, there is Omicron or Omicron, which has another name, BA1. And then the newest strain of the coronavirus, which is circulating in the world, including starting to increase in numbers in the United States, is BA2. And that might be BA.2 or DA period 2, I'm not sure, but BA2. Okay. BA2 is derived from BA1. BA1 was derived from Delta virus. I don't know the history of where Delta came from. There are other different strains. These are the strains that cause epidemics in the United States. Any question about any of that? They all have different mutations. And when you get infected by one, like if you were infected with the original or alpha, I think beta too, you are not protected against BA2. BA2 is different enough that if you got the original, you will have no protection from the original. Unfortunately, the vaccine is based on the original COVID-19. And so BA2, the vaccine will not protect you to the BA2 strain. What the protection is, we don't know because BA2 is too recent. For the protection for BA1 or Omicron, the protection was under 50%. And I think it was around 35%. I used to know this, I don't remember what it was. I do remember what Delta was. If you got the vaccine, you were protected 65% of the time from Delta virus. They're hoping that the vaccine, even though you will get BA2 in most of the cases, over 50%, probably over 70%. You will get the virus BA2, meaning COVID-19, if you have been vaccinated, but they are hoping that the vaccine will reduce the severity of the disease. So far, at least with BA1, if you get the vaccine and you get BA1, your chance of dying was much, much less, and the severity of the disease was much less if you were vaccinated than if you got the disease or the BA1 virus. Any question about any of that? All right, here's a question for you. Delta virus was the most lethal strain of COVID-19 that we know of, at least so far. It killed the highest percentage of people who became infected. BA1, or Omicron, was the least lethal, the least severe. All right. So if BA1 was the least severe, why are there more deaths from BA1 Omicron than there were deaths from Delta, which had the highest lethality rate?
Is it because people were practicing precautions to not get Delta, but with Omicron, a lot of people weren't?
Yes, that's exactly it. Although there's another reason you can state too, and that is people were not taking precautions with Omicron because they heard it is less lethal, less severe, and it is less severe. It's in fact, this one, as far as we know, was the least severe COVID-19 strain. We don't know about BA2, but I think BA2 will be about the same lethality and severity as BA1. We don't know because it's so new, meaning they need to study it a little while longer. Maybe we'll know in a month. But BA1 or Omicron was less severe, but people weren't taking precautions. So a lot more people got infected with BA1 than they did with Delta. And then Delta also had a much longer duration than BA1. This one came and went quickly, but a lot more people got BA1. And yes, it is less severe, but more people died of BA1 because so many people came down with BA1 or Omicron. So yes, it is less lethal, but people are still dying from it. That's my point. And so you do need to take precautions. And like I said, the deaths in the United States are now over 1 million cases of people dying from COVID-19. BA2 is the strain of the virus currently going around in the United States. They think it's going to increase this spring, and hopefully it'll decrease before the summer comes. BA2 is currently the most transmissible COVID-19 strain. It's 1.5 times more transmissible than BA1, which used to be the strain that had the highest transmissibility. But BA2 is even more transmissible than BA1. Any question about that? I think the original was slightly more transmissible than the flu. And as you know, the flu can very easily spread from person to person. BA2 is much more transmissible than the flu. And I'm talking something like, it's something like 20% more transmissible than the flu. And it might be as high as 50 times more transmissible than the flu, because I don't know the exact numbers. I only know hard fact that it's 1.5 times more transmissible than BA1. And I think BA1 was about 10 times more transmissible than the original. Any question about any of that? All right. The transmission of COVID-19 is primarily via droplets from coughs and sneezes within a range of about 6 feet, but you can spread it simply by breathing. It is known that singing tends to spread COVID-19. So don't sing around other people, especially if you're sick. It is transmissible from spreading from indirect contact, meaning if a surface or something becomes contaminated, you can pick it up on your hands and then spread it to a mucous membrane of your face. COVID-19 is also found in feces, but we don't think it spreads this way. COVID-19 is likely breathed in. For whatever reason, it appears to rarely be picked up by your hands and then carried to the face. This is unusual because with the colds and flus, most of the time we pick the virus up in our hands, and then we inoculate ourselves in a mucous membrane of the face. COVID-19 is known that it can survive for 3 days on plastic or a metal surface, but it only survives for 1 day on paper or cardboard. So one thing you can do to protect yourself is when you get the mail, let it sit for a day and then you can open it. Okay. Any question about any of that? The symptoms, it's mostly flu-like, though the runny nose and the sneezing are less common. I think with the later strains of COVID-19, they are becoming more and more like the flu, meaning the runny nose and the sneezing are more frequent. The shortness of breath and deaths are more common. The mortality of COVID-19 increases with your age, with the ill health of the patient, meaning if you're already ill and then you get COVID-19, you're more likely to die. Deaths and severity are more common in men. Pregnancy increases the severity and the deaths of COVID-19. And for reasons that I've never heard anyone explain, Blacks and Hispanics have more deaths and more serious disease than whites and Asians. Any question about any of that? All right, the incubation period, the time before you become infected till when you first have signs and symptoms, are 2 to 14 days from when you're first infected to where you have any signs. Some people are asymptomatic. Most of you are young adults, young adults, teenagers, and children, it's estimated that about 50% of you, when you get infected with COVID-19, you will be asymptomatic. And that is actually how it got spread to the United States. Because if you remember when COVID-19 was first coming around, they, at the airport, first of all, they didn't let people from China come into the country. And then they were checking people to see if they were sick at the airports and at the shipping docks, trying to keep COVID-19 out of the country. But an asymptomatic patient brought it into the country. And if you're young, 50% of the time, you'll be asymptomatic. The estimated death rate, as I mentioned, was between 0.1% to 3%. The actual death rate that they've measured as of May 31, 2022 is about 1.2% of people who become infected in the United States of COVID-19 will die. Now, the death rate will vary depending on the strain, but all of the strains do have a death rate. Okay, Delta having the highest death rate. Prevention, use correct hand washing. When you wash your hands, you should make sure the soap goes everywhere, and you should wash everywhere, as well as wash for 30 seconds. Keep your distance from people. That way you can't pick it up. Avoid people who are coughing. And then don't touch your face, especially if you've touched things. Don't touch your face until you wash your hands or sanitize your hands. If you're sick, please wear a mask. That'll prevent the spread of the disease from you. It's best to stay home and then cough into your elbow or your tissue. Any questions about any of that? All right, let's briefly talk about viruses and cancer. Some human cancers, about 10 to 15% of human cancers, are caused by viruses. Some examples: human papillomavirus causes cervical and skin cancer. Hepatitis B virus and hepatitis C can cause liver cancer. Hepatitis is a liver virus, so that's why hepatitis means liver. Not all viruses cause cancer, and not all cancer is caused by a virus. For example, Tang et al. Found in 2013 that breast cancer and glioblastoma in humans is not associated or caused by cancer. Any question about any of that? There are two main ways that viruses can cause cancer. You can have a virus forming a provirus in the DNA of our cells, where the virus integrates into the chromosome and it permanently genetically changes the cell for the lifetime of that cell, where the DNA virus and retroviruses integrate into our cell, and that can mutate the cell, leading to cancer. Any question about any of that? The second way a virus can cause cancer occurs in an oncogenic virus. These viruses contain an oncogene. An oncogene is a gene that is a normal cellular gene involved in cellular growth, and that normal gene becomes mutated and then causes the cell to grow abnormally. That mutated normal gene we call an oncogene. And the virus, when it infects the cell, can cause that cell to start growing abnormally, which can lead to cancer. Any question about any of that? So there are two ways the viruses can cause cancer. One is the virus can bring in a gene which can lead to the formation of cancer, and that gene we call an oncogene. And the second way is the virus can have its genome integrate into our genome, and it can muck up our genome, and that can lead to cancer. Any question about any of that? If not, let's talk about latent and persistent viral infections. A latent viral infection is where the virus remains in the patient, but the patient is asymptomatic and will be asymptomatic for long periods of time. Generally, the latent virus does not reproduce. It just sort of sits there doing very little. It does not cause disease while it's in latency. And then for whatever reason, the latent virus can come out of latency, activate, and then start replicating. This happens with herpes simplex virus 1. When an individual has a cold sore, the virus is actively replicating, and it's going through the budding life cycle at this time. And then after the cold sore goes away, the virus stops replicating, leaves the budding life cycle, and enters the latent life cycle, where the virus is just sitting around in the cell in latency. Another example is a child comes down with chickenpox. The chickenpox virus is actively replicating, causing the disease chickenpox. And then after the chickenpox goes away, the virus leaves replication, and whatever life cycle that is, I should look that up, I don't know. I think it's the budding life cycle, but I'm not positive, so don't quote me on that. And it goes into the latent life cycle, where the virus just sits around in a nerve cell, a ganglion nerve, and it sits around, it can sit around for decades. And then the virus leaves the latent life cycle and enters the replication life cycle, and the patient will have disease, but it won't be chickenpox. Every other time the patient has a disease, we call it shingles, caused by the same virus as chickenpox. The first time you get it, it causes the disease chickenpox. Every other time you get disease from that virus, we call it shingles. There is usually a trigger for activating the virus from the latent life cycle to the active life cycle, causing the shingles. It's usually a stress, which puts stress on the immune system, and then the virus comes out and replicates. With the cold sore, the stress is usually a cold. And that's why we call it a cold sore, because people tend to get cold sores when they have a cold. Any question about any of that? Let me briefly talk about persistent viral infections. This is a disease that occurs over a long period of time. The detectable levels of the virus build up over a long period of time. So initially, when the patient is first infected and they first have signs of disease, the virus is at low number. But over time, the viral load increases and the severity of the disease increases. The virus oftentimes is never cleared by the host immune system. The disease may be fatal, such as subacute sclerosing panencephalitis, which is caused by the measles virus. And then a better example that everyone's familiar with, HIV can be fatal, causing a persistent viral infection, which we will call causing the disease AIDS. And if the patient dies from AIDS, it was a persistent viral infection. Persistent viral infections do not have to be fatal, even if they're lifelong. Many of them are tolerated, like a cytomegalovirus or CMV. About 50% of Americans have this virus, not 50% having a persistent viral infection of this virus. But my point is, this is common in the United States, and many of these viral infections are tolerated. Varicella zoster, which causes chickenpox. My mother actually had a case where the shingles never went away. She always had the shingles, and she just tolerated it. It is true that she never had the virus stopped replicating, but she never got rid of the shingles. And then there are other herpes virus that sometimes cause persistent viral infections. When we look at a graph, looking at the persistent viral infection, I need to shrink that a little. There we go. And this will be the number of virions as well as the severity of disease. With a normal viral infection, which we call an acute infection, the patient becomes infected. The viral load after incubation, the viral load then increases very rapidly, coming to high numbers, and then the virus decreases very rapidly. Like the human cold follows this. We get very rapid increase in the virus load, we get sick, and then the virus goes away. That's an acute infection. The latent virus would be something like chickenpox. The child becomes infected with chickenpox, and then they go better, but the virus is never cleared. It goes into latency, and then many years later, you can have another infection that we call shingles, and then the virus load can come out. You can have shingles more than once, okay? But that's a latent infection. And once again, it's similar to the acute infection. You have a viral load increase rapidly, maybe not as high, and then it decreases rapidly. Persistent infection, once you become infected, you have a very low load initially, and this load increases, and then you have severe disease near long time after you become infected. And you notice how this breaks? That's because the patient died. You don't always have the patient dying, so it can plateau out here. All right, any question about persistent viral infections? Prions, let me just briefly talk about this subject, and then we'll finish about prions next time. Prions are proteinaceous infectious particles. These are believed to be proteins that cause human disease, where the proteins are infectious. They can be inherited, meaning someone can inherit a gene which makes a protein, which then leads to prion disease. And then you can transmit the disease by ingestion or transplant or what do you call that, inject it into the patient, or surgical instruments can bring the prion to the patient. Prions tend to cause spongiform encephalopathy, where it makes the brain have vacuoles in it, which causes holes in the brain. I've got a picture of it here, which makes the brain look something like a sponge. Right here and here and here. There's the normal brain there. And that's where the term spongiform encephalopathy means. Sponge, meaning it looks like a sponge. Encephalopathy we won't talk about. This is a disease of humans and animals. In animals, we call it bovine spongiform encephalopathy, more commonly called mad cow disease. There is another disease, which is scabies, a disease that European farmers have been fighting for centuries, meaning living with for centuries. The disease of sheep, and another case, cervix chronic wasting disease. We'll talk briefly about mad cow disease next time. In humans, we don't call it that. We don't even call it human spongiform encephalopathy. We call it Creutzfeldt-Jakob disease, a variant of Creutzfeldt-Jakob disease. Gerstmann, Strauss-Lauer, Schneckner syndrome. These are named after the people who discovered it. And then fatal familial insomnia and Kuru. And all of these diseases are caused by a prion. Any question about any of that? If not, I'm going to stop here and we'll talk about this next time. All right, any question about anything? All right, no questions. I will see you next time. If you have questions about the lab, I'll be there from 6:30 to 6:45.